Hello friends, this is Dr. Pankaj Kumar, and in this session we will be talking about embryology, right? We know very well that once the fertilization is going to happen, zygote will form and zygote will immediately undergo the very process of embryological development. So it undergoes the very process what we call cleavage. So first of all, we will discuss what exactly is a cleavage. So it undergoes a series of rapid mitotic division, okay? So eventually which convert the single cell zygote into multicellular structure, and the structure formed what we call it is a blastula. So what happens first of all, the first cleavage is going to happens after 32 hours of fertilization, forming 2 blastomere, okay, and the second cleavage occurs after 40 hours forming 4 blastomeres, right? The third cleavage after 3 days of fertilization forming 8 cell structure, okay, and at the end of 4 days what happened, by that time embryo also reaches the uterus and it has 8 to 16 blastomere, okay, and the solid mass of cell that is known as morula. So when the blastomere divide completely, the cleavage is called holoblastic, right? So let's see blastocyst formation. So what happens in an embryo with about 64 cells, a cavity is formed within the cell mass. This cavity is actually called as a blastocells. You can see in the diagram. So this is the blastocells, okay. So what happens, it consists of an outer envelope of cells, the trophoblast, and the inner cell that is called embryoblast. So this is the outer what we call trophoblast, and this is the inner what we call embryoblast. So the site of blastocyst to which the inner cell is attached is called embryonic or animal pole, right? And the opposite is known as aembryonic pole. Now what happens, trophoblast encircle the blastocells and the inner cell mass, right? As we can see the diagram. The cells of the trophoblast later on form the extraembryonic membrane, right? This will form extraembryonic membrane, okay. And the cells of the trophoblast which are in contact with the inner cell mass are called cells of Rauber. So these are contact. So actually it is not shown in the diagram, but when these are contact, this is actually called as the cells of Rauber, right? Let's talk about implantation. You know, implantation is going to occur after 7 days of fertilizations. About 8 days after fertilization, the trophoblast develop into 2 layers in the region which is in contact with the blastocyst in the endometrium. What are these layers? These are syncytial trophoblast that contains a non-distinct cell boundaries, and cytotrophoblast between the inner cell mass and the syncytial trophoblast that is composed of distinct cells. We can see these things in diagram. Right now we have learned that outer wall is called trophoblast, this is called inner cell, right? And this is actually in contact with the endometrium, okay. Then within we see there is a cell, this is what we call cytotrophoblast, okay, between the inner cell mass and syncytial trophoblast, and that particular structure, this structure is actually called as a syncytial trophoblast, right? Now what happens that the cells of the inner cell mass differentiate into 2 layers. A layer of a small cuboidal cell known as hypoblast, and a layer of high columnar cells known as epiblast. So these are actually called as a epiblast. Both the hypoblast and epiblast form a flat disc structure called embryonic disc, okay. So these are together is going to cause as a embryonic disc. Zona pellucida prevents the implantation of the blastocysts at any other level and any other abnormal site, right? Because it does not expose the sticky and phagocytic cells of trophoblast till the blastocyst reaches the proper implantation sites, okay. So it is the function of zona pellucida that ensures the very proper implantation of the fertilized product, right? Now let's try to understand that how we can differentiate between fetus and embryo. In human beings, what happens? that the developing organism from conception till approx at the end of 8th week, it is called embryo. And beyond the 8th week, the fetus is the unborn young one of the viviparous animal, okay, and it has taken form in the uterus. Now we start call after 8th week we start calling them the fetus, okay. So the status remain fetus from 8 weeks onward till the birth, right? Now let's talk about the gastrulation. What exactly is a gastrulation? So what happens? There is a transformation of the blastocyst into the gastrula with a primary germ layer. There are 3 types of primary germ layer: ectoderm, mesoderm, endoderm. These are the 3 primary germ layers. So in case of gastrulation, what happens? There is a morphogenetic movement of cells is going to happen. And when cells are moving, so as a result of movement, what happens? These layers eventually lead to the very process of organogenesis. Okay. Now have a look at some of the primitive stage of gastrulation. The very first, what we call formation of the embryonic disc. The hypoblast, we have already discussed, is a layer of the columnar cells, and the epiblast is a layer of the cuboidal cells. They together form an embryonic disc. So we can see there, this is together what we call embryonic disc. In the previous diagram also we have seen that. Now this is followed by the formation of the amniotic cavity. Now what happens? A space between epiblast and trophoblast called amniotic cavity filled with amniotic fluid is formed. Have a look at this diagram. So this was the epiblast, okay, and this is the trophoblast. Then epiblast and trophoblast, then this is the amniotic cavity that is going to be formed, and that is filled with the amniotic fluid, okay. Now the roof of this cavity is formed by the amniogenic cells derived from the trophoblast, while the floor is formed from the epiblast, which is very much clear, okay. Then there is the formation of extraembryonic coelom. What happens? Cells of the trophoblast give rise to a mass of a cell called extraembryonic mesoderm. Later, these extraembryonic membrane develop into amnion and chorion. Now flattened cells arise from hypoblast, spread and line inside blastocells. So these are the endodermal cells lining the primary yolk sac. Okay. Now there are 4 extraembryonic membrane that is formed, what we call chorion, that covers fetus and other membrane, amnion that forms protective fluid filled sac around embryo, yolk sac that is primarily found that is more prominently developed in case of lower animals, allantois that used to remove the waste material that is formed during the morphogenic or the developments, and umbilical cord that is that provides food and oxygen to the developing fetus. Now have a look the same thing in the diagram as well. You can see there. So this is the chorion, that is the outermost layer, outermost protective layer, right? And this is the white color structure. What you see, it is a allantois is present over there, okay? And then this is the amnion and this is the amniotic cavity. So the fetus or is actually floating within the amniotic cavity, okay, or within the amniotic fluid, right? Now finally, after implantation, what will happen? A finger-like projection appear on the trophoblast called chorionic villi, which are surrounded by the uterine tissue and the maternal blood. The chorionic villi and uterine tissue jointly form a structure and functional unit between developing embryo and maternal body, and what structure we call placenta. So we can say placenta is made up of as many as 6 layers, 3 from the fetal side and 3 from the maternal side. So the very function of the placenta is to facilitate the supply of oxygen and nutrients to the embryo. Placenta is connected to the embryo through umbilical cord, which helps in the transport of substances to and fro from the embryo. Now you see, placenta also act as an endocrine tissue and produce hormone like human chorionic gonadotropin (HCG), human placental lactogen, estrogen, progesterone. So these are the things that is secreted by the placenta itself, and they have a role in maintenance of the pregnancy and thereby the other role, right? So these are the entire things as far as the entire embryological development is concerned. After this, what will happen, that over the period of time, as per the months proceed, then the organogenesis is going to happen. That is beyond the scope of your syllabus, so that's why we are not talking about. At the different period, different organs developed, and eventually after 9 months, the baby is going to be born, right? That's all as far as the entire embryological is concerned. Okay, thank you.